Our team chose to genetically modify algae to produce proteins that will purify water sources around the world.
The new balance in the water will create a healthier habitat for the animals that live in the water and the animals and humans that drink from the same water source. This allows for poorer third world countries to obtain a source of fresh water instead of having to drink the water that is contaminated with millions of bacteria that can lead to disease, infection, and in worst cases, death. In the year 2006, The Guardian reported that there is an estimated amount of 5,000 child deaths per day due to a lack of clean water. Now imagine a world where every child has access to clean water without having the fear of getting diarrhea, cholera, or any sort of life-threatening bacteria in their water supply. That is something we can all strive for, as everyone deserves to drink clean water.
The significance of our project is that it manages to clean water from bacteria without any limitations. We chose this topic because as of right now, 1 in 3 or 2.4 billion people lack the access to clean water. Every 90 seconds, a child dies from a waterborne disease. Access to adequate water, sanitation, and hygiene is critical in the prevention and care of 16 of the 17 neglected tropical diseases, including trachoma, soil-transmitted helminths, intestinal worms, and schistosomiasis. NTDs affect more than 1.5 billion people in 149 countries, causing blindness, disfigurement, permanent disability, and death. These are all very alarming statistics, and that's why we chose this as our topic. There has been many attempts on treating water turbidity and bacteria before. One of them was when a biocoagulant was successfully extracted from Plantago orata by using an FeCl2-induced extract. It was tested on the turbidity of a river. After several experimental tests were performed, results demonstrated that the FCE removed more than 95.6% of all initial turbidity concentrations. Our project is very different from any others that have been attempted before. Our project uses genetic engineering to its utmost level by inserting a protein from an algae to the bacteria and ultimately coagulating it. Other treatments on water turbidity are usually inorganic coagulants and synthetic organic polymers. While their other projects work best in water at pH level less than 8, our project does not have any such restrictions. To begin our project, our gene of choice will be inserted into the ACT2 gene of the algae.
The ACT2 gene is the place where the insertion of the MO2.1 gene will occur. The ACT2 gene is found on the first chromosome of algae and encodes for actin-related filaments, which are proteins that help support the cell. The MO2.1 gene is found in the horseradish tree. The important part of this gene, though, is the proteins that it produces. The protein coagulates water. The process of coagulation is when there is sediment or other small particles in the water and they are removed. These particles are usually negatively charged, so to neutralize them, a chemical that is positively charged is added. The horseradish tree naturally creates a positively charged protein particle that neutralizes the sediment in the water. This gene is what we want to insert into Chlamydomonas reinhardti, which is a single-celled algae. The promoter that will be used is the HSP78. This promoter will be good for the gene expression of the MO2.1 gene because the promoter can use either one or a combination of Mg-proto, heat or light. Mg-proto is magnesium protoporphyrin 4, which is a molecule that can mimic normal light induction. This will provide for constant gene expression, especially since most of the countries with a dire need for this bacteria is around the equator, meaning they are constantly hot and the sun shines pretty much all the time.
The incorporation of the MO2.1 gene will occur because the restriction enzymes locate the specific DNA sequences in the gene, cut them, and then the complementary base pairs of the foreign sequence attach in these spaces. Once the foreign DNA is in the ACT2 gene of the algae, it will produce proteins that coagulate and fluctuate bacteria and water impurities. Now that the algae will start to filter out bacteria and water, it will create a favorable balance for the water system it is in. The scientific technique we chose was to incorporate a plasmid into the genome of the algae. The plasmid is from a horseradish tree, and it produces proteins that coagulate water contaminants, which is an incredibly helpful trait to our genetic engineering project. Another component of the plasmid will be the KPNI and BAMHI restriction enzymes. They recognize and create sticky ends with specific DNA sequences in the ACT2 and MO2.1 genes. KPNI recognizes the sequence GGTACC, while BAMHI recognizes the sequence CTAG.
To successfully incorporate the MO2.1 gene, it will be inserted into the genome of Chlamydomonas reinhardti. Since this is an algal organism, there is generally only one cell type. This species mostly exists as a single-cell haploid organism. So these are the cell types in which we will be inserting the MO2.1 gene. After DNA transfer, though, there must be a method to measure the effectiveness of the transformation. A few specific properties of the MO2.1 gene present an opportunity in which quantitative analysis of the transformation can be done. The specific gene encodes for proteins that coagulate water contaminants and bacteria. A water sample that has been treated with a genetically engineered algae can be analyzed, and the data can be collected for the amount of contaminant coagulation as a percentage of the total amount of water. Through these analyses, the effectiveness of the transformation can be measured. Continuing with this, after the water is allowed to interact with the algae and flocculation of bacteria has occurred, the created clumps can be removed from the water. To test whether bacteria was removed or not, the water can be plated on a dish with an agar medium. This dish will be monitored for bacterial growth, and if no bacterial growth occurs, then the engineering was completely successful and all of the bacteria was removed by the algae. Though it would be ideal to have a completely effective DNA transformation, this almost never happens. One technical hurdle that may be encountered is the precision of the restriction enzymes. The restriction enzyme, KPNI, was chosen, and it only targets one specific DNA sequence in both the chromosome and the gene. On the other hand, BAMHI is able to recognize a sequence that occurs in the chromosome 6 times. Because this sequence occurs several different times, it's possible that the chromosome will be split in a different spot than intended. To combat this, though, the team chose 2 restriction enzymes that have clustered restriction sites. This limits the negative effects of the restriction enzyme, because the restriction enzyme will most likely find one of the 4 closely packed sites and cut the chromosome there. On a more grand scale, another negative outcome that is possible for this project is that the algae could be unable to survive in the turbid water they're placed in. There may be too many water contaminants, or the water could be too cloudy to support plant life. This would directly limit the effectiveness of the engineered algae. If the organisms are unable to survive, they would be unable to perform their intended function of purifying the water. We hope the world will be benefited by our genetic engineering project, and we thank you for watching.